So for histology, I don't talk about this a lot, but there are images available for each lab that are going to have mostly histology. Usually I'll point them out in the virtual videos if I think they're super helpful. So there are some slides like in the digestive system, for example, that there's a ton here and it can be overwhelming. So I still think the best resources are going to be the histology stuff. I think that's the main stuff I will use in the videos as well. Okay, so we're going to be covering everything from cardiac through the reproductive systems. So if I just go down the labs here, I can kind of tell you what will be emphasized. There is going to be, there'll be slides about blood vessels. We'll have just usually one or two questions about blood histology itself. So that's going to be more like a blood stain or a blood smear where we have erythrocytes and everything else. So you need to know, especially things like neutrophils and lymphocytes, the more common blood histo there. There will be stuff from respiratory, urinary. There will be a lot from GI tract, so we'll spend a lot of time there, and then also a little bit from the most recent virtual lab. So we'll go ahead and work down the list here. The Human Protein Atlas is also really good, but it doesn't have every single system. So that's going to be more relevant for things like the respiratory system or the digestive tract, less so for blood vessels and blood itself. So do we have blood histo? Okay. So I think the thing that mixes students up the most for blood histology is just identifying our leukocytes. So let's pull up. This is a little too zoomed in for what I want. Sites. So this is a little further magnified than we can get with our slides, but this is what you can expect for a blood question. Most of what you'll see by far will be erythrocytes. So the hollow centers here indicate that these are going to be erythrocytes. They have that biconcave appearance because they don't have nuclei. There's not a lot going on in the cell. That's most of what you'll see. And we can't forget about the background here. So these red blood cells are going to be suspended in the fluid portion of blood, which is going to be plasma. So we'll see a ton of that. Usually you'll have a couple platelets scattered around, so those are also fair game, but they're pretty hard to locate on a microscope, so that might be more of a PowerPoint question. Now that gets tricky when we look at, where'd it go? When we start to look for those leukocytes. And you might want to refer back to the attendance worksheet for blood histo, because that one had you fill out a chart. And it also talked about the frequency, the prevalence of all of these within a typical stain. Usually, you'll either see red blood cells or neutrophils. Those are by far the most common types. Sometimes, if we're lucky, we can find a lymphocyte. So the lymphocytes are going to look a little different. They usually just have one lobe of a nucleus versus if we switch back to neutrophils, you can see we usually have 3 to 5 lobes within that nucleus. That's like the main way I would tell apart these on a slide. You can look at the appearance too, like granular versus agranular, but I think the best way to identify these types of cells is just counting the lobes of the nucleus. So between those two, those are usually the only ones that I can find on a slide within the time I have to set up the lab exam. So hopefully that clues you in to what to study. And then we can move to blood vessels. So for blood vessels, I had you guys look at that microscope slide that has arteries, veins, and nerves on it. And that is kind of what to expect for histology of the blood vessels, that there's not going to be any sort of identification of specific vessel names. It'll be more of that looking at the layers of the blood vessels, trying to compare differences between arteries and veins to identify them. So, and some of these examples were used from the Yale site. I'm going to go ahead and pull up a couple different types here. And I'm really going to keep it to arteries and veins. I could ask you about arterioles or venules, but that's a little smaller scale. We want to focus on the most important things, which is going to be veins versus arteries. All right, so this is going to be the thickest artery with the thickest possible tunica media. So we have an aorta wall here. It will have the same layers. So one important thing to remember is that our layers of the blood vessels will be consistent for both arteries and veins, even though they look a little different. So with things like the aorta, or especially elastic arteries that are closer to the heart and do more of that job of withstanding high blood pressure, they're usually going to have a lot of elastic fibers. And those don't show up super well on this slide, so I will also pull one up from our modules so we can see it better. But typically we'd expect to have a lot of elastic tissue in arteries, elastic arteries. So let's go here. Oh, that's too far. So this is going to be a different perspective. In this case, the slide has been stained to show those really dark elastic tissues. So if you see a lot of dark squiggly lines like this, that is going to be indicative that this is an elastic artery, versus arteries that move further away from the heart are more of those muscular arteries, which we have right here. So there's really an absence of elastic connective tissue within that tunica media, that middle layer. Don't let that mix you up with the internal elastic tunica interna here. So usually all of the vessels we talk about will have just a little bit of elastic tissue, and that'll be found in that innermost layer. That's part of what creates these squiggly patterns in the layer itself. However, we don't have that same pattern of a lot of elastic tissue in the tunica media. So that's going to be the main difference between muscular and elastic arteries. And veins are going to be very similar, where they don't have a lot of elastic tissue, and that's what gives them that floppier appearance. So we've kind of mentioned two layers. You can't forget about your tunica adventitia. That is going to be more of that connective tissue layer towards the external part of the vessel, so more superficial. And if we compare that to a vein, it's probably not the best example, so let's find one here. So this is a great slide showing those comparisons. What we have on the right is an artery. One clue is that we have a round lumen. So arteries, because they have more elastic tissue, are going to retain that round shape much more easily than the veins. In this case, this looks like an elastic artery because it has a lot of darker stained tissue, which clues us in that it's elastic tissue. Veins are going to be much floppier. So just by looking at the shape of the lumen here, you can see it doesn't hold that round shape. It's not pressurized because we don't have that same blood pressure as blood is returning to the heart. So there's no need for that elastic tissue. What else? Those are pretty much the main things to know. Does that make sense so far, vessels?
Yeah, that answered most of the questions. So just focus on identifying elastic versus muscular, the three tunicas,
Yep, and just
and
arteries
just
versus
veins versus artery. Yep.
Right. All right, let's see what is next. Respiratory. Okay. Let's go ahead and look at respiratory. So this one has some tricky histology. The models themselves are not going to be too horrible because you've got the lung model and the bronchial tree. Those are the main ones. The histology can be tricky though. Oh, I don't know if I mentioned this before, but if you're looking on the Yale site and you're looking at slides here, there are also virtual microscope slides. So we might look at that one as well. That is going to be a lot more similar to what we do in class, and where I have you set up the microscope and then poke around. So this is also a really good way to study. And it does have these labels. Some of them are going to go beyond the scope of this class. Yeah, so probably don't worry about the labels, but you can use these blank slides to practice too. So histology-wise, things that were mentioned would be things like the trachea with that pseudostratified ciliated columnar epithelium. That is pretty much a catch-all phrase for the respiratory epithelium. That is not as likely to show up. So I'm going to focus more on testing your understanding of the alveoli, the actual microscopic level at which gas exchange is happening. So that side is going to look a lot more like this. So we are within the lung tissue. This is going to be that transition from the conducting zone to the respiratory zone of our respiratory system. So at this point, after entering the lungs, we have the primary bronchi branching off into tertiary or secondary and tertiary. At this point, we're dealing with bronchioles and then the air sacs themselves, the alveoli. So the main way to tell those things apart is our bronchioles are still going to have smooth muscle associated with them. The reason for that is we need a way to dilate these vessels when we need to increase our air intake. And there's also a way to reduce dilation when we don't need as much air supply. So that's going to be related to like a sympathetic versus parasympathetic response, fight or flight. But that's why we have this smooth muscle so that it can contract and dilate depending on other signals. And the squiggly stuff you see here, that it has more cellular tissue, that's smooth muscle. So we're going to see patterns forming of either like a cross section of looking down on a tube lined by smooth muscle, or we will also have passageways. Let me try to find a good example. Maybe down here. So you also see stuff like this where we have a passageway forming, but it still has the smooth muscle around it. I say all that to say that this is a terminal bronchiole. So if you see smooth muscle, you're dealing with a bronchiole. Everything else on here, once that smooth muscle goes away, we're dealing with alveolar structures. So the main unit of air exchange is going to be one of these little clusters. These are alveoli, and they are lined by cells. In this case, those cells are going to be pneumocytes that are either producing that lubricating fluid, or they're going to be the actual cells allowing gas exchange between blood and these alveoli. So that's all very microscopic, but this unit is going to be an alveolus, sorry, that word's hard to say, alveoli. And then sometimes you'll see these clusters of air sacs connecting to a tube. So maybe in this case, we've got one alveoli here, we have a larger passageway. So you'll have alveolar ducts that are eventually connecting back to things like our terminal bronchioles. So this slide is a little messy. I'm going to try to find one in the modules to compare. But we'll still
So
be
most likely
we
we would probably just be asked to distinguish between like an alveolar and a bronchiole on a slide like that.
Yes. Yeah. So if you see, like, for example, if you see a round cluster of cells like this, that's going to be an alveolus. Alveoli is plural. If you see this same structure, but it's more of a duct pattern, like instead of having clusters, you'll have like a long passageway. That's an alveolar duct. And
Okay.
then the kind of the last thing to know, as you mentioned, is the terminal bronchiole has that smooth muscle lining it. Okay, so here's a really nice example. So each of these individual units is again those air sacs. They're going to connect to an alveolar duct, which is what we see here. There's no smooth muscle around it, so that tells us it is going to be an alveolar duct. And then eventually that will lead back to this bronchiole, which has the smooth muscle. Does that make sense?
Yeah. Yep. That's good.
Okay, cool. How do you feel about bronchial tree?
If we could go over that and the maybe like a picture of the lung model as well.
So this is basically where our lungs will be. So the point of entry to the lungs is going to be these primary bronchi. So this is what is hidden by our lung model. Overall, this is one continuous passageway for air, and that's important to remember because we're going to have things like tracheocartilage lining the entire thing. Some of that cartilage can also be seen under a microscope. So I didn't mention that. It's not super important for identification, but you may also see that on a slide. We can't forget about the larynx. So we did gloss over a lot of the smaller structures in the larynx, but you do want to know things like the thyroid cartilage, which is that most superior kind of shield-shaped cartilage. That corresponds to our Adam's apple. So that's that protuberance you feel, that's actually our thyroid cartilage. Just underneath that, we have the cricoid cartilage, which I like to think of as C-shaped. So cricoid starts with a C, it's C-shaped, so it's curving around to the back, and it gets thicker as it curves to the back. And then everything else here is going to be those tracheocartilages. Once we get to this level here, this point where the trachea becomes the bronchi, the splitting off is called the carina, C-A-R-I-N-A. So if I put an arrow right here on that point of the division, that's carina. And then we have our left and our right primary bronchi. And make sure you say bronchi, not bronchiole. Bronchioles are going to be the little ones that we see like in the histology. So left and right. And then we get into the secondary bronchi. And I like to use the naming system, primary, secondary, tertiary. That makes more sense to me than trying to remember like lobar and that kind of stuff. So primary is going to branch off to secondary. So those on this model are still covered by a little bit of cartilage. That's one way to tell them apart from the tertiary bronchi. Same thing on the right side. And then the colorful bits here, those are going to be our tertiary bronchi. And those are the last bronchi before it starts branching off into bronchioles. Theoretically, you can count, so you'll have 2 versus 3 in the lungs in terms of secondary bronchi, but I'm not going to ask you that on the lab exam. It'll be mostly just identification of primary versus secondary versus tertiary. And then there's one more thing that we can't see. So if you flip this model around, you can see the epiglottis sticking at the top. So I also like to include that one because it's important to recognize that the epiglottis is going to fold over our air passageway when we swallow, so that food and drink don't go down that way. So that is all bronchial tree. And then we also have our lungs. Okay, so this one is going to be more superficial structures. So really basic stuff like the number of lobes per lung and identifying the names of the lobes, as well as these fissures that we see in the middle. So left side is just going to have that superior and inferior lung. The way we can tell which is left versus right is we also have this cardiac impression. This is where the heart is going to rest. And it's very obvious on this structure, which is nice. So make sure you know that impression. We also have fissures. So the left side is going to have just that oblique fissure. Right side is going to have oblique as well as horizontal. So make sure you know those differences. I don't remember if I asked left versus right lung, but you can use the cardiac impression to identify left versus right. But make sure you're solid on your fissures as well as names for the lobes of the lungs. That's
So
pretty much
for like
all.
labeling,
Uh-huh.
so for like labeling the lung, so say like you put it on the left, like superior lobe, would we have to
Yep.
say like left lung, superior lobe, or would it just be like superior lobe?
Um, well, it's multiple choice, so I don't remember if
Oh, okay.
I probably did specify left superior versus right superior, but you'll have it as an answer choice. Okay. All right. Let's see. Let's have GI tract and then we'll do urinary, since that one should be more fresh for you guys. GI tract is going to show up a lot because there are models I can ask that from and lots of histology. And this is probably the most confusing lab for students because it can be hard to tell apart the GI tract at the different points, like the stomach, the small intestine, and the colon. I'm going to work backwards, actually. So we'll start with the colon because the main way to tell the colon apart from the other regions is the presence of lots and lots of goblet cells. So similar to the blood vessels, the entire GI tract is going to have the same layers, just with a couple variations throughout, especially in the mucosa, that innermost layer. So that is where we see a lot of variation that we can use to identify the different regions. In this case, the colon has just tons and tons of goblet cells. Yes, we do see them throughout the entire GI tract, but they're going to be most prevalent here in the colon. So if you see this pattern with lots and lots of light goblet cells, that should be your clue that we're dealing with the colon or the large intestine. Let's see if we have a different one. Yeah, this is a good PowerPoint question as well. Okay, so this slide, I'm probably not going to emphasize this on the lab exam, but this slide does also show that repeating pattern of goblet cells in light. What it also shows is the Taenia coli, which is going to be those strips of longitudinal muscle running along the outside of the colon that are creating that kind of roughly appearance. So the three bands of muscle, the Taenia coli, run all along the expanse of the colon. They're a little shorter than the actual colon, so those are what are creating the little pouches, the haustrum. So let's see if we can find that on our model. Let me try this one. Wrong model. No, okay. So maybe I didn't post a model of this. If you watch the video, the virtual video for GI tract 1, I go over the large torso model with all of the digestive tract. And on that one, you can also see things like Taenia coli. So be careful with this one. It is visible from this microscopic perspective as well as on our model. So that's going to be colon. And then we can use the Human Protein Atlas to look at some parts of the digestive tract. So let's start with that small intestine. All right. So this one, I believe, combines two parts of the small intestine. On our slide, I don't know if you looked at this in class, but for our microscope slide, we have 3 separate circles of tissue from each part of the small intestine. So microscopically, you do need to know the difference between duodenum, jejunum, and ileum. When looking at the macroscopic model of the torso, you do not need to know those separate regions. So that's going to be solely identification under a microscope. So let's start with the more proximal part of the small intestine. This is the part that is interacting with the stomach. So once food is exiting the stomach, the first part it enters is the duodenum. Again, we want to look towards the mucosa or maybe a little bit into the submucosa to identify these layers. So one thing you will find throughout the entire small intestine is the presence of plicae circularis, which are those really big, really deep finger-like indentations here that are just maximizing surface area for the exchange of nutrients and waste. So you're going to see that entire pattern all along the tract as we move down here. But if we focus in on the mucosa, we start to see these glands here. Lots and lots of glands. These are Brunner's glands, or I think the PowerPoint referred to them as duodenal glands, though these are only found in the first part of the intestine, the duodenum. Their function is to help neutralize the acidic contents coming out of the stomach. So it makes sense that we would have these closer to the stomach, but not have them further away as we move down the tract. So that's your main clue that we're dealing with duodenum, is the presence of those glands. Usually they're housed in the mucosa or maybe just a little bit into the submucosa, but you want to look for them in the mucosa. We're going to skip over the main part of the small intestine for now and talk about the most terminal portion. And the tricky part will be finding our unique characteristic. Okay, so this is the terminal part. In this, we have Peyer's patches in the ileum. So these guys are actually made of lymphatic nodules. So functionally, they're very similar to lymph nodes. They kind of look, they have the shape of a fingerprint. That's what I look for when I'm trying to identify them. But all they are is just this round cluster of lymphatic nodules. Those are solely found in the ileum, that terminal portion of the small intestine. You can see another little one over here. And they will look similar on our slides. Usually they are a little darker stain and they're going to have that round kind of grainy appearance to them. And then lastly, we have the middle part here. And let's see if we have a nice slide here. Maybe. Okay. Let's see what Yale has. Sometimes we have to flip back and forth to find good slides for the jejunum. Okay, let's try one more thing. Jejunum. There we go. Okay, so jejunum is identified by process of elimination because there are no special characteristics. It's kind of the basic unit of the small intestine. So we still have plicae circularis, this really windy, large surface area structure. What we don't have is Brunner's glands or Peyer's patches. So you'll have to use process of elimination for this one. Usually I do try to give clues. For example, I might say, identify this immune structure within the small intestine, and you'll have to know what that is based on what it looks like on a slide. A good follow-up question might be, what region of the digestive system or what region of the small intestine is this structure found in? So we can use those unique characteristics. With the jejunum, it's more identification because we don't have anything really special about it. Does that all make sense so far? I know we flipped back and forth a little bit.
Yes. So like I'd say probably like big picture, looking at like you see the slide for the first time, you have to narrow it down to at least like small intestine. So you know you're working with the duodenum and jejunum and ileum. Probably just look for villi, and then from there start looking for those individual like the unique glands and cells.
Right.
To narrow it down to which specific part of the small intestine.
Yep. So always look for that big picture stuff first, and that'll help you orient yourself. I could also ask about layers here. So I could ask about mucosa, submucosa, or the muscular layer, muscularis externa. But we also have models for that. So that's where this guy comes back. This is our generic small intestine model. So we still have that pattern with lots and lots of villi increasing the surface area. And once you recognize that, you recognize these as the villi, that will make your life a lot easier. So this is going to be the layer closest to the lumen of the small intestine. So all of this is going to be in the mucosa. There's a lot going on. You don't have to know individual vessels or glands here, none of that. Just recognize the regions. So this is going to be that innermost mucosa. Next layer down, you can see it gets a lot busier here. So we have a lot more of those glands, many, many vessels, lymphatic and green, and then we have arteries, or most likely arterioles and venules. in red and blue. This layer is submucosa, and it's mostly a combination of connective tissue. So it's not as dense, but it does have a lot of vessels running through it. And then our last layer is our muscularis externa. We'll have two layers, so longitudinal and then intercircular. And yeah, I would go ahead and know the difference between those two based on the direction of the fibers. The outermost layer is going to be longitudinal. So that is how I would orient myself. Find the innermost lumen layer and then work your way outwards from there.
You said the two layers of the muscularis externa was longitudinal and horizontal.
Yeah, let's see. I swear, everyone has a different name for them. I don't remember in the PowerPoint if we used horizontal. Let me check. Where's my PowerPoint? Okay. So I'm gonna make sure I'm giving you the information we have from the PowerPoint. Keep moving down here. There we go. Okay, so we do use circular. So we're going to use circular for the inner and then longitudinal for the outer one. Different textbooks are going to have different stuff, but those are going to be the answer choices that you have. Don't get that confused with oblique layer. There is an oblique layer, but it is only found in the stomach. So let's go ahead and finish out with the stomach. Yes, the esophagus and the oral cavity are part of the digestive tract, but we're not really going to focus on those for the lab exam. Okay, so here we have our stomach. This is the space, the lumen within the stomach. So it's pretty not distended right now. It's actually really relaxed, showing some of those folds, those rugae within the stomach. If we zoom in, we start to see our individual layers. So again, we've got mucosa, a little bit of submucosa, which is usually lighter stained, and then we're going to have a really thick muscular externa. And this is where we still have that outermost longitudinal layer. We still have a circular layer, but then we have this really thick oblique layer. And that is unique to the stomach. It's what allows it to churn material together to form that chyme and start that mechanical and chemical breakdown of food. Other things that are unique to the stomach, there's one more thing in the mucosa here. So we'll have, they almost look like crevices. I think of them as crevices. We'll have these really narrow passageways called gastric pits. You can see this example here. It's just a really deep crevice going pretty far into the mucosa. At the end of each of those gastric pits, there will be a gastric gland. Those are going to be secreting things that help with the highly acidic chemical breakdown of food. But I'm most likely going to ask about the gastric pits because they're a little easier to see than the glands. So if you see the arrow pointing to one of these slits here or here, really they're all over, that's going to clue you in that this is the gastric pit in the stomach. Yeah, so between those two, between the oblique layer and the gastric pit, those are two unique characteristics I could ask about in the stomach. All right. Anything else you want to hit with the GI tract? There's also accessory stuff.
I'm looking at the lab attendance. Looks like we just went over salivary glands, the pancreas.
Yes. So I'm actually going to pull up that PowerPoint too. I'm trying to make it real simple for you guys with the accessory stuff. Otherwise, salivary glands can get really complicated. So let's find that PowerPoint because there's a really helpful slide in here that I used to study when I took this class. So let's find. Here we go. This slide. So there are 3 salivary glands. You need to be able to identify all of them when you're just given a slide. So there's not really anything I can put a pointer on that's going to help you or clue you into which gland is which. So you just have to know the distribution of serous versus mucus cells in each of these glands because they all look slightly different based on color and kind of the clustering of cells. So probably the easiest one to identify is sublingual. I think it has the most distinct appearance. So sublingual is going to be mostly mucus, which is why it's lighter stained. Typically, mucus cells show up light stained. That's why the colon had all of those little dots of really light goblet cells that are making mucus. So that's what we see here. It is a mixed gland, so it's going to have usually these kind of berry-shaped clusters of those mucus cells surrounded or capped by darker cells. That's some of what you had to do for the attendance worksheet. Sublingual is, oh, I mixed them up. Sorry, I meant to say sublingual. I think I said submandibular. Yeah, sublingual is the lightest one because it has most of those mucus secretions. I was referring to submandibular, so that was my bad. So submandibular is going to be the mixed one, which if we look a little closer. Yes, okay, so submandibular glands are going to be that mixed pattern. So if we zoomed in a little bit here, we would start to see this kind of repeating structure over and over again. So what we're dealing with is these berry-shaped clusters of cells. They're called acini or acinus for singular. And so this is what creates that kind of mottled pink-white pattern in the submandibular gland. So it has these lighter stained mucus cells capped by these darker serous cells. And that's what we see up top right here. We'll have those lighter patterns kind of outlined in dark purple. So that's going to be the mixed gland. Again, apologies for mixing that up. Sublingual is going to be that mostly mucus, so it's the lightest of the bunch, and then that leaves parotid, which is the darkest stained. So it's going to be mostly those serous secretions. Does that make sense?
Yeah, yep. The three
Okay.
differentiating the three is good.
Yeah, so you're mainly just looking at color there.
Okay.
And I do like to clue people in. So if you know which cell or which glands have which types of cells, that is going to help you answer the question. So make sure you know serous versus mucus and which are found in which. Big hint there. All right. Oh, I did want to mention on our liver model, this guy is weird. So it's not an anatomical position, which is why a lot of students tend to mix up left versus right. So I want to make sure we cover that today. First, let's go ahead and orient ourselves. That's super important for this one. So the big vessel here in blue is inferior vena cava. It's the main passageway for that venous return to get back to the heart. That is always going to be posterior in anatomical position. So this tells us this is posterior. Gallbladder is going to be more anterior. And then we are also looking at this from the bottom up. So we're looking at it from like the perspective of the diaphragm, which is not at all anatomical position. So this is going to be the left side based on what we know. This is at the back. We're looking at it from the bottom. So this is left in anatomical position, and this is going to be our right lobe. That one just takes time. So just be careful when you get to the liver model that you recognize this is a deviation from anatomical position. So once you orient yourself, left and right becomes really easy. Quadrate lobe is going to be this quadrangular shaped guy, and it sits right next to the gallbladder. So I just go ahead and study those two together, try to associate them. And then that leaves the smallest lobe, which is the caudate lobe. One way that you might remember this is caudal refers to tail. So this is kind of towards the tail and the posterior end of the body. Let's see. I don't think I really ask about blood vessels from this view. Mainly because we have the blood vessel man. We have that usually just a couple questions about the names of arteries and veins. And these can all be asked from that view, which is an anatomical position. So I would focus mostly on identifying lobes Know that this is the gallbladder, and then does not featured on this model, but we usually will have a white structure right here, which is the falciform ligament. So I know the model we use for the test will have that structure on it. What that looks like is it runs right between the right and the left lobes. So from this kind of inferior posterior perspective, you can see a little bit of that falciform ligament. So we would expect to have it right here. How do you feel about liver histology?
If we could probably just go over it from the top.
Mhm. So there is a liver plaque and there is also a liver slide. So the plaque is a little more zoomed in. What we have on the slide is the main kind of unit of the liver, which is the liver lobule. That guy is roughly hexagonal in shape, and all of them are always going to have a central vein. So actually, this reminds me a lot of when we talked about compact bone, where we had that unit of the osteon with a central opening, a central canal. And this is very similar. In this case, we have liver lobules with a central vein. And the liver itself is going to have a lot of really tiny vessels called sinusoids that are draining towards that central vein. So it's a really efficient drainage system. This slide also shows our portal triad. You don't need to recognize that for the slide, but that is similar to what we see from this view, where we've got our hepatic artery, portal vein, and then we also have a cystic duct. So those three kind of bunched together on the sides of our model. What we can see on our plaque here is we have individual liver cells. So in each of the round units in the middle are going to be nuclei. These are called hepatocytes. So hepato refers to liver, smash it together with cell, and you get hepatocyte. And then we'll also have those central veins in the middle. So some things that you can see on both, like the central veins and that portal triad. Other things are going to be unique to each, like the hepatocytes versus the liver lobule. This one is really going to be orientation. So if you recognize the shape and recognize this is liver tissue, and you also know this is a central vein, it should be no problem. But if you don't know that pattern, this is going to be a lot harder to identify. Let's see. I know this picture here is available in our modules. It shows some of the specialized cells of the liver. This would definitely be more of a PowerPoint question because we can't really zoom in that closely on a slide. But what we have is so we have eosinophils, but we also have lots and lots of Kupffer cells. And these are going to be doing a lot of the filtration of the liver. They actually line the sinusoids, those tiny vessels, and they're going to be just doing a lot of that phagocytic filtration. So you don't need to know the function. It would be helpful to recognize that these are Kupffer cells. But other than that, you don't have to worry about specific cells for the liver. Let's see. Okay. And then the last one for accessory is the pancreas, and this should hopefully be review. We did see the pancreas before when we talked about the endocrine system. So, and you're going to use the same structure to orient yourself to the pancreas. What we'll have is islands of those endocrine structures, which were the islets of Langerhans or the pancreatic islets. So if you recognize those on your slide, then you should be able to identify this as the pancreas. From there, the exocrine tissue that we're concerned about for this lab is going to be acinar cells, not to be confused with the salivary glands. So these cells are really just secreting, storing, producing those pancreatic juices that will eventually join into and dump into that pancreatic duct. So I can see a lot of students confusing this with salivary glands. So what you'll need to do is recognize this islet of Langerhans. If we took those away, this would pretty much be a salivary gland, just with a different function, but they would look almost identical. So we need those to identify this as pancreas.
So like I could absolutely rely on seeing one of those islets of Langerhans on like on a slide that we would see on test.
Right.
Like I wouldn't
Yep.
have to. Okay, gotcha.
Yeah, I'll make it fair. Because it's so similar to so many other tissues, we really need those clues to identify it. Okay, I think that hits digestive tract. That was the main area I wanted to really tackle because of the histology. How are you feeling about renal system? I know that was a lot.
Yeah, if we could hit that, that'd be great.
Yes, so we need 2 slides here. Actually, this will work. So there are 2 slides that I can ask this about. These are specific to the kidneys. So while it is important to recognize all the structures of the renal system, so everything from kidneys and their blood supply, ureters, urinary bladder, and urethra, that's all good to know from that macroscopic view. For histology, we're really just focusing on the kidneys. So our model will have, or our microscopes, sorry, will have the entire kidney. And I was trying to find a slide here, but I don't know if it is available. We can work with the Human Protein Atlas. So what you'll have, this is like half of a kidney here. What you'll have is the whole kidney from one of our slides. You will be able to see the hilum as well as the cortex and the medulla. Actually, just pull it up right now. So from that macroscopic view, you want to be able to identify the layers of the kidney. Here we go. Okay. So like I mentioned a lot, we have many organs with an outer cortex and an inner medulla, and the kidneys are no exception. So the darker part here is going to be that outer cortex. Technically, there is a fibrous capsule, but that is usually kind of separated on the slide, so don't worry about that. And then we have the inner medulla, which is pretty thick. On our plaque, we can see the renal pyramids or the medullary pyramids that would be all along the medulla here. And then lastly, we have that theoretical opening called the hilum. This is the entry point for our artery, our vein, as well as the renal pelvis, where it connects to the ureters. So that's the theoretical entry point. So you might see this slide crop up. That would just be asking about regions. Some clues that you can use to identify regions, or some wording I might use are, I might mention most of the filtration happens in this region, which would be the cortex. A lot of blood vessels are found in the medulla. So you might see those types of clues that you can use to identify the regions. The other slide we are dealing with is a microscopic view of glomeruli. So I mentioned it over and over again in class, but the glomeruli are going to be the filtration units. This is where blood is being taken to the kidneys. It's certain things are being pushed across a pressure gradient and filtered out, and they form that filtrate. Blood gets taken back out of the kidneys, and that filtrate continues its journey to the ureters, where it eventually becomes urine. That's kind of a 20-second summary of the renal system. If we zoom in on the cortex here, the outermost part, we start to see these round structures. These are glomeruli. So what we have is a corpuscle, a renal corpuscle, which is this entire unit. Inside, we have a bundle of capillaries called a glomerulus. That is the actual site of blood and filtrate exchange. So what it looks like is a big old mess. You'll see red blood cells here. We've got a lot of repeating capillaries, and they're usually just lined by one layer of cells to increase that exchange of gases and nutrients. You will have some specialized cells as well, but you don't need to know those from this slide. I just want you to know glomerulus. And then we also have this space around here, almost like a moat between the glomerulus and the surrounding tissue. That is that Bowman space. So sometimes you'll see the renal corpuscle referred to as the Bowman's capsule or the Bowman space in this case. But you can use those two ideas to identify it. Find your round unit with this little moat. This is the Bowman space, and then the thing in the middle is the glomerulus. How that connects with the big picture is filtration is happening up here. We're going to have parts of the neuron, maybe that loop of Henle dipping down into the medulla. And then eventually we'll have collecting ducts making their way from the cortex through the medulla towards the hilum. So that's kind of how this system works. And we'll have just lots and lots and lots of nephrons throughout the entire kidneys. You might see things from that view. You will definitely also see things from our plaque. Trying to see if we have a picture of the whole thing. Maybe not. So the plaque had 3 views. We have the macroscopic view of the kidneys, which looks like this. And then we have the middle part, which shows an entire nephron. And then the last view, which is not on this PowerPoint, is the smallest view, just showing a glomerulus. But spend a lot of time with this perspective. So just to orient ourselves, this is going to be part of the inner medulla. This is the renal cortex. And the way we can confirm that is glomeruli are found within the cortex. So each of these round units is a renal corpuscle. It's going to have that fibrous outer capsule and tan. This guy here has been chopped in half so that we can see into the corpuscle and see that glomerulus. And connecting to it, we have that entire system of tubes, the renal tubule, which is altogether forming this nephron, this unit of filtration. I think a lot of students get the tubes mixed up because they have really weird names. And if you don't think about what the name means, it's easy to mistake them for each other. We have basically really twisty tubules, which are called convoluted tubules. They are going to be either really close, very proximal to the glomerulus, or they'll be all the way over here at the collecting duct. So this whole thing is collecting filtrate from all of these glomeruli and sending it off towards the ureters. So start at your glomerulus when you're trying to identify anything on the nephron. Start here. The first structure you encounter is the proximal convoluted tubule. It's proximal to the glomerulus and it's really convoluted. It's twisty. On the opposite end, we have the distal convoluted tubule. So again, it has that same twisty structure, but now it's distal. It's closer to that distal collecting duct. In between, we have this loop. So the order matters. Start with the glomerulus. You go through your proximal convoluted tubule. You're going to descend. So this is the descending loop of Henle. And then you'll go back up. So now we're ascending. So this is our ascending loop of Henle. You get back up to the twisty bit, which is the distal convoluted tubule. And from there, everything is filtering into the collecting duct. Does that make sense?
Yeah, yep, that's good.
Okay, yep, just start at the glomerulus, whatever you do to identify this stuff. Otherwise, people get into trouble with proximal versus distal, ascending versus descending. What else?
So on
Don't forget
that,
about, oh, go ahead.
so on the kidney plaque with the 3 sections, the furthest right one, that's only the glomerulosa.
Yes.
So like, would we be asked to identify like a structure on that, or like just identify what that is?
That one, it could be either. So on our model we use for the lab exam, you can see podocytes. So this view is kind of messy, but I just want to point out we have podocytes wrapping around our, oh, here we go. We have podocytes wrapping around the actual capillaries of the glomerulus. So I could put an arrow on this and ask you to identify this specific cell. These guys are what are forming those filtration slits that kind of force fluid across the pressure gradient to form that filtrate. So they're important to know. So your model might look like this, where it has some capillaries exposed and some covered in podocytes. So make sure you know the difference between those two. Other things we can see here would be the Bowman's capsule or glomerular capsule. Same name, same structure. We also have arterioles, which now that I think about it, I don't think I test you on because they're easy to mix up. So probably just focus on the capsule, glomerulus, and then the podocytes wrapping around individual capillaries. And then other thing, don't forget about blood vessels from this view. So most, like by far and large, blood vessels will be asked from either the blood vessel model with the man covered in arteries and veins, or from the heart. Things like our great vessels are good heart questions. But there are a couple cases where we have some arteries and smaller arterioles to pay attention to. So that's why we spent a lot of time last class going over this view, and we talked about the sequence of arteries. So you don't need to memorize this entire sequence, but if given a view like this, like on our plaque, you need to be comfortable identifying the larger stuff here. So things like renal vein, renal artery, segmental arteries that are branching off to different pyramids, basically everything on this scale up to arcuate arteries. And it's all fair game. Okay, if you're cool with that, we can, let's see what else we need to hit. Go back to Yale. I think all that's left is reproductive. And then if you would like to, we can also go over some structures on the heart model. Okay. Just going to refer to the PowerPoint for this one so everything's in the same place. There we go. Okay. So for reproductive, where people get in trouble is orientation and histology. So I won't spend too much time on the models because that's part of what the attendance sheet was working on. But everything is going to pretty much be asked from this mid-sagittal view for the female and the male reproductive tracts. I don't think people have trouble telling apart the male and female, but they get in trouble with all of these cavities here. So from this view, we have stuff from the renal system. So we have the bladder. We have stuff from the digestive system. So this is rectum and anus. And then we have reproductive stuff. So a lot of people will confuse these other openings with things like the uterus, for example, on the female model. In males, the bladder is going to sit more anteriorly, and it sits anterior in terms of the rest of the view that we see here. And it sits above the prostate gland. So that is one way to help identify and orient yourself here. Bladder sits above everything, whereas in the female model, this kind of view, the bladder is going to sit more forward, so it's more anterior. Same deal here. A lot of people will confuse the uterus with the bladder or vice versa. So it's important to know the bladder sits more forward in the female model and it sits above stuff in the male model. And then this, of course, is going to mirror rectum and anus. So for whatever reason, people will confuse the tubes here. I usually try to keep it pretty basic. So I'll ask things like the big structures, uterus, vagina, ovaries, fallopian tubes, anything that was mentioned in the video and kind of hammered home there, it's fair game. But people still miss this because they get messed up. They see all these openings and get confused. So I would just practice orienting yourself, making sure you know your know anterior versus posterior and what the heck the bladder is doing in each of these systems.
So if I see like a mid-sagittal view model, so it's like those aren't limited to only reproductive, like labeling, like everything's fair game.
It will be,
like as far as structures.
If you see this, if you see this view, it will be a reproductive question.
Right.
But people get in trouble because they think like the bladder is the uterus. or the urethra is the vagina. So just use those things to orient yourself, but all these questions, if you see this kind of model, they will be reproductive.
Okay, gotcha.
All right. Okay. I will say ovaries are one of the most complicated slides that we deal with in this class because there's a lot of concepts that overlap here. So big picture stuff, the ovaries have an outer cortex and an inner medulla. We've seen that pattern over and over. I spend a lot of time in the videos talking about stuff in the cortex because this is where we find follicles, which are producing the female gametes, which are well, eggs, ovum. So I recommend rewatching that video because there are many stages of follicles. So just to give a brief recap of this process, it is called oogenesis. It is the production of a mature egg cell, a mature ovum. There are a couple of different stages along the way in these ovarian follicles. So this is the site of that production and maturation for the egg cell. Before the egg is released during ovulation, it is referred to as an oocyte. So oocyte. And that is what is going to be contained within all of these follicles. The default state for an ovarian follicle is going to be primordial. And this actually all occurs before birth. So this is true that the eggs are, the number of eggs in an ovary are determined before birth in the female reproductive tract. What that is referring to is the structure contained within the primordial follicle, and that is called a primary oocyte. And those terms are easy to keep together because they sound similar. Primordial goes with primary. These follicles are all in kind of the default rest mode. So up to a certain point of cell division, the primary oocyte will form, and then that process is going to halt until puberty. So nothing is going to happen to these oocytes until puberty, when ovulation starts to occur. So that is the rest mode. That's what people were referring to when they talk about the fact that eggs are predetermined before birth. In terms of what it looks like, it is pretty basic. So it's just a round unit with just a single layer of squamous cells. And in the middle, you'll have a primary oocyte. So this is a slightly zoomed in version. So the stuff in the middle, oocyte. Stuff on the outside will have just a thin layer of that squamous tissue. Then we get this primary follicle. So you got to wait several years, and then post-puberty, it will start to develop into a primary follicle. And the main difference in appearance is that the primary follicles have cuboidal cells surrounding them versus squamous. So we've gone from just that really thin outer layer to a slightly thicker layer. So you can start to see you can see a noticeable difference between the oocyte and the surrounding cellular layer. The main difference between primary and secondary is we just have more layers. So secondary, you're still dealing with cuboidal epithelium, but in this case, there's just a lot more layers. So this is primary follicle. It just looks like one layer of cuboidal cells. You can start to see those cells are expanding, and then we have this additional structure kind of encapsulating the whole follicle. This is not testable, but that structure is called a theca. It looks like just a little kind of membranous border around that secondary follicle. So the thing in the middle is still a primary oocyte, so that has not changed throughout this entire development until we get to the tertiary follicle, which is that last stage of maturation for the follicle. So this looks a lot different now. We've gone from a cell with kind of a membranous, or not a cell, a follicle with this membranous theca and lots of layers of cuboidal epithelium, and that has just exploded now. This is much larger. We still have those layers of cuboidal cells, but they've been pushed way out. And in the middle here, we have this cavity called an antrum. So definitely want to know that term. In this case, within that mature tertiary follicle, this is now called a secondary oocyte. And that secondary oocyte is what is released during ovulation. So this follicle is actually going to rupture to release that secondary oocyte. And then it'll undergo some changes post ovulation. So it'll get smaller and it'll shrink down and kind of disappear if no pregnancy occurs. So that is kind of the 5-minute recap for all the stuff you need to know about the ovary. And all of that is occurring within this cortex. So it's a very busy area of the ovary. Feel okay about that?
Yeah, feel good about that.
Okay, good. That's a good slide, good microscope question. So make sure you're really solid on those. And then there's really just 2 other slides for the entire histology of the reproductive tract. And those are male tract slides. Okay, so the testes, and you can't see this on the model, the testes have these really squiggly patterns all throughout the tissue. What that looks like on a microscopic view is this. So this is a cross section. What we have done is we've taken a cross section of the testes. So we're looking down now on these many tubes. And so this is the lumen of a tube. This is another tube right next door. You can see that pattern over and over again on your slide. These are seminiferous tubules. So these are where sperm are going to be formed. There are several types of cells that are helping to nourish those sperm cells, or they're also producing sex hormones. So we see some of those surrounding the seminiferous tubule. Nurse cells or Sertoli cells will be found in here. Don't worry about identifying those under a slide. That's just extra information. But they are nurturing and nourishing these sperm cells during development. You will see a couple other types of cells. So in between the tubules, we'll have these Leydig cells, which are producing testosterone. And then you'll also see these cells that look kind of like smooth muscle that are just going to contract those tubes. But I would say the main thing to know is when given a hint that this is like male reproductive tract or the site of sperm production, you want to be able to recognize that this is a seminiferous tubule and this is found in the testes. And then the last slide to focus on is the epididymis. So the testes and the epididymis work closely together. Testes are producing semen, not semen, sperm, sorry. And then the epididymis is the site of maturation and storage. So they're just, think of them as one continuous vessel. We just have different names along different points here. And the epididymis looks similar, but there is one difference to help you identify it. And that is the presence of stereocilia. So if you look closely on the inner border here, we have these hair-like projections, and those are those stereocilia, the hair cells. What they do is help move the sperm along. They help transport it out of the testes and then eventually into the vas deferens. And that is really one of the few ways to tell apart this tissue. So if I were to give you a slide like this, I would make it so that you can identify those stereocilia pretty easily. Right. If you're okay with that, we can wrap it up with cardiac. I think that's our last one.
Yeah, I'm good with repro. Yeah, cardiac, that's about it.
Let's see. We need some heart model views. Also, don't forget about the dissection of the heart. There will be a couple questions. And they'll be pretty basic. So maybe great vessels or pretty easily identified structures of the heart, but we don't want to forget about that. This is a good view. So there are 2 views that this model can be used from. We can see something like this where we have the internal structures, or I could put the other half back on and ask about great vessels, or maybe those superficial structures like the auricles. But this is probably the most confusing view, so we'll focus on this one. So you need to know right versus left. Things that will help you are the angle of the heart. So if you recall it is angled slightly towards the left so that the apex of the heart, this pointy part, is pointing slightly to the left from anatomical position. So if you remember that, you can identify left versus right chambers. Other clues, we can look at the wall thickness. So the left ventricle is responsible for pumping blood to most of the body, everything except the pulmonary circuit. So in order to do that, we need really thick muscular walls. And so that's what you can see here on the left ventricle. Compare that to the right, this is a lot thinner. And this difference is even more dramatic in those hearts that we dissected. So you had a really big left ventricular wall and a much thinner right ventricular wall. In between, we have an interventricular septum. And so that's a good one to put an arrow on. You want to be comfortable with muscles, so things like trabeculae carneae and the corresponding muscle in the atrium. Those have different names, and students tend to mix them up. Be comfortable with valves as well. Since it is multiple choice, you want to be able to recognize both names for the valves. So we talked about bicuspid and mitral. We talked about atrioventricular. So we'll have a left versus a right atrioventricular valve. And then there's also the pulmonary and the aortic valves. So those are smaller. They're going to be tucked up here. You may also see them referred to as semilunar. So that's 4 terms now for 2 types of valves. So make sure you know left versus right are really solid on that. And then you also know the different names for all of those valves. I'm not really going to ask questions about the order of things. So it is helpful to think about how blood is, for example, returning from the systemic circulation to the right atrium and right ventricle. It's helpful to know this order, but the questions are going to be strictly identification. Things that get confusing are the pulmonary circuit, I think, and just knowing when blood is oxygenated versus deoxygenated. And the color coding here can help you with that. So the blue is generally going to be venous structures. We have superior vena cava returning blood to the right atrium. Atrium is separated by ventricle by this valve. So we have right ventricle, left ventricle over here, left atrium is hiding away up there. You want to definitely know your great vessels. So in addition to superior and inferior vena cava, we also have this aortic arch with those 3 branches in alphabetical order, left to right. We have pulmonary trunk. This is a tricky one for people. So it has a purple color. It's different. People sometimes forget whether it's an artery or a vein. So we need to think about the blood flow. Right ventricle is pumping blood out towards the lungs. Because it is leaving the heart, this is going to be an artery. So sometimes you'll see the pulmonary trunk splitting off into the left and right pulmonary arteries. Let's see if we can find a posterior view of the heart. So for the pulmonary circuit, we'll just switch back and forth here. This is now a posterior view of the heart. So we still have that aortic arch with the 3 branches. Inferior vena cava is now on the right. So this is right side, left side for an anatomical position. So that pulmonary trunk is going to wrap around and split off into the left and the right pulmonary arteries. And then we have our pulmonary veins. And so the colors are switched now because once blood goes to the pulmonary circuit, it's going to get filled with oxygen from the lungs. And then that oxygen has to come back to the heart so it can be pumped out to the rest of the body. And it returns to the heart through these paired pulmonary veins. So don't let that confuse you. These are red because they're taking oxygenated blood back to the heart, but they're still veins because we have those different circuits. What else? Lastly, you don't want to forget about what is anchoring the valve in each of these ventricles. So we have the valve itself, but we also have the heartstrings, the chordae tendineae, which are these stringy white bits, and they are connecting to a papillary muscle. So these nubs here are papillary muscles. They are different than the surrounding trabeculae cardiae. Now, one thing you will notice is not on the model here is all the electrical stuff. So we did briefly talk about how an electrical signal and heartbeat is propagated throughout the heart. It's not on the model, so you will not be tested on it.